2 hours ago at Heathrow, is that right? And hot.
I arrived 10 minutes ago.
Right, okay, but your flight only landed about 2 hours ago, I'm guessing.
Yeah.
So Arnie is Professor of Biology and Director of Skeletal Research Centre, Case Western Reserve University, and a good friend of mine, I must say. And Arnie, to many of us in this room, is one of the guys who we looked up to when we started. You are the godfather of MSCs, or the father of MSCs. So would you mind just giving us a potted history of MSCs and your role as a scientist who came up with the term, named them, and actually then formed the first major company in the space, Osiris. So if you could just talk a little bit about that.
So actually it's all quite logical. I was working with embryonic chick limb mesenchymal cells and developed a way in the late 60s, it kills me to say that, late 60s, to take the cells from a limb bud of an embryo, put them in culture and study their muscle, cartilage, and bone development. And because we had a media in which we expanded these cells, when I started working with adult mesenchymal stem cells, that was the magic media, and it was hugely lucky at that time because the whole technology is controlled by the batch of fetal calf serum that's used in the media. The sequence of events actually was that my really good friend Marshall Yurst worked for many years on trying to purify the BMPs, and when Genetics Institute cloned the BMP, John Wozni and his colleagues, I realized, even though I had 3 patents on BMPs, that I had lost the race for the IP for BMPs, and the assay that we had used was to take embryonic chick limb mesenchymal cells, put them in culture, and under conditions where they would never form cartilage, and take an extract from demineralized bone, purified extracts, put it on the culture and show that the whole culture goes to cartilage. And so we had an assay which we called chondrogenic stimulating activity. You can look up the patents because they're totally worthless currently. And when Genetics Institute cloned the BMP family, I knew that my lab had lost the race. And then I realized that Marshall Yurst had stuck a hunk of demineralized bone in an adult muscle pouch, and from that, de novo bone arose. And so from that observation, I said, there must be mesenchymal stem cells there of some sort. And let's go find them. And I had this assay, and of course I had been involved in orthopedics for 40 years, and the orthopedic surgeons from the days of Aristotle have used marrow as an additive to all of their bone reconstructive surgeries. And so it had this magic juice in it. And so I figured the magic juice may be these mesenchymal stem cells. I was completely ignorant, I have to say, of most of the Friedenstein and the early literature on adult osteogenic cells, but I was a good friend of Maureen Owen. Maureen Owen was a professor of orthopedics at Oxford, and first of all, a woman, second of all, in orthopedics, a guy's business. And Maureen had written an enormously clever review article in 1985, and she had a figure in there. The figure was the hemopoietic lineage cascade, and then she sort of scribbled the mesenchymal cascade. And she didn't get the whole picture, but she got it, that there were in marrow these progenitor cells that could certainly make bone and fat. And she had good data for that. And she had sort of rediscovered Friedenstein, who was in Russia and mostly undiscovered by the West. And so all of that came together at the same time. And Steve Hainsworth and I went after human MSCs, and for 6 months, every every single experiment we did not only worked, but worked better than anybody could have planned it, even if they knew there were MSCs there. So to say that we had a horseshoe appropriately placed is an understatement. And so I started a company which was called Skeletech.
Mm-hmm.
So Jim Burns, the first CEO of Osiris, thought that that was not a Wall Street name, and so therefore it was like skeletons in my closet. and so he changed the name to Osiris, and there's a bunch of funny stories about the name which I'll spare you of. But in the end, Osiris was started in December of 1992 as a bio-orthopedic company to tissue engineer replacement parts. So we didn't have a clue as to what MSCs could do in reality and what the current science of MSCs is, which is, for me, quite spectacular. And every day I come into my lab and there's a new something going on, and it's quite exciting.
So that was very much to use them as their differentiated product, as bone or cartilage or fat, whereas today we seem to have moved them more towards their proangiogenesis.
No, it turns out it's both, because there's still a good logic for using MSCs in reconstructive surgeries. The issue is probably this is going to be the basic science that we've developed for cartilage and bone in particular, probably is going to be superseded by embryonic stem cells and or iPS cells, because these cells, you can get gigantic numbers of cell divisions and get the number of cells that you need to tissue engineer human-sized tissue. Tissues for mouse and tissues for rabbit, no problem. All the horses in this room, your knees and hips are huge, and it takes gazillion cells, and probably adult MSCs from human are going to be okay for some things, but eventually 10 years out, the capacity to differentiate and expand iPS cells and embryonic stem cells are going to supersede this.
So is that using them as a starting material to make MSCs, or is this using them to derive the therapy you want? I mean,
I think both. I think, actually, as a point of disclosure, I'm a consultant for Mike West's company called Biotime, and Mike showed me the data several years ago, which enchanted me to become a consultant in his company, is that he has clones derived from embryonic stem cells that he can expand forever, and these clones are chondrogenic. And so, for example, he's trying to now road test a variety of those clones in hyaluronic acid specific scaffolds for cartilage products, and so he has a full program going with that, and that's one of several programs, and again, they're tissue engineering programs quite separate from what we're going to talk about now, which is the medicinal capacity of these cells. And
in fact, you prefer a different name for them, isn't it?
Yeah, so I invented this name, mesenchymal stem cells, to be provocative with my esteemed orthopod colleagues and to get them to think in a new way. And actually, when I would lecture originally, I would say, don't call them mesenchymal stem cells, call them Caplan cells. and because I took an enormous amount of heat from my esteemed colleagues whose lips I could read, and they would be saying BS. You have to remember, most of you are, half of you are young enough not to remember that in the late 80s, there was only one adult stem cell, and that was the hemopoietic stem cell. So now we know there are kidney stem cells, liver stem cells, neurostem cells, heart stem cells, etc. Those are committed progenitors which are involved in the actual continued regeneration of those tissues. And as it turns out, the MSC is the custodian of the activity of those cells in a regenerative context. And so the whole saying that there was a mesenchymal stem cell was heresy, complete heresy at the time, and it changed people's thinking. And then eventually, thank goodness for Mr. Bush and his lack of intelligence, by banning embryonic stem cells, he made mesenchymal stem cells quite popular, because we had adult stem cells and we didn't have the ethical problem that Mr. Bush and his colleagues had with embryonic stem cells.
But you've been naming them, was it medicinal, producing, sorry.
Yeah, you know, I have a delicate ego, so I want everybody to continue to use MSC as the most important way to describe these cells, but their activities, which I'll describe in a minute, have absolutely nothing whatsoever to do with stemness. And so I've renamed them medicinal signaling cells because they're drugstores for sites of injury. They function naturally at these sites, and all of the activities are very site-specific, and there is, and again, the MS, you know, I worked with marrow my whole life, and the concept that you could get MSCs from fat or placenta. It's just the most disgusting idea. I can't imagine these precious and incredibly powerful cells coming from fat or this tissue that you throw away, placenta. But the bottom line is that every single blood vessel in your body has pericytes on it. These pericytes are the vascular responsive elements. So when Chris takes his blood pressure medication in the morning, these are the guys who are sitting around his blood vessels that are responding. When you want high blood pressure, they squeeze, and when you want low blood pressure, they relax. And when that blood vessel is injured or inflamed, the MSC comes off. The pericyte comes off and becomes an MSC. Then from the front of that MSC, it secretes a curtain of molecules that absolutely stop your overaggressive immune system from surveying the damaged tissue behind it. This is your first line of defense against autoimmune diseases. And from the back of the MSC, they produce molecules which inhibit scarring, inhibit cell death, and actually enhance the slow process of regeneration of that tissue. So they are the managers of each of your innate regenerative capacity. And that's why, if you go to clinicaltrials.gov, this website, you'll see there's 400 clinical trials currently in play on clinicaltrials.gov, and almost 95% of them have nothing whatsoever to do with tissue engineering. They're all the medicinal aspects, and all of the clinical symptoms have two very important components. One is they have an immune component for which the MSC is regulatory, and the second is a regenerative component, destroyed tissue which regenerates.
Now, Arnie, just to pick up on that, because I think that's a really good point. I mean, being slightly provocative, I mean, we see a lot of MSCs out there. We see ones from Mesoblast, the Exosiris ones, yours, of course, the Athesys cells, and many others. And you say, 400 plus clinical trials. Are these all pretty much the same cell? Are we looking at a class effect here?
All of the IP for all the companies is wonderful and indistinguishable.
Absolutely.
And completely challengeable in every sense of the word, because an MSC, whether it comes from brain tissue, from skin, from fat, does these functions, seeking out sites of injury, setting up an immune barrier, and setting up a regenerative microenvironment. The amazing piece of this is that given the 30 or 40 companies that are culturing these cells, they're not destroying any of that activity. So everybody's secret formula, everybody's method of isolating and culture expanding these cells, and Athesys, a favorite company of mine because they're in Cleveland, they culture cells for 100 days at low density and low oxygen in the presence of growth factors, conditions that most of us find revolting. And the fact that they take them through so many population doublings, and yet those two capacities, the immunomodulatory, for which they have fabulous experimental data as well as clinical data, and the regenerative capabilities are still intact in those cells. And although my good friend Catherine Prof invented those cells for other reasons because they were pluripotent, not just multipotent, again, the medicinal capabilities of those cells, which are derived originally from real MSCs, that capacity is maintained. And so that's the take-home message. Here is a capacity in a group of cells that's hardwired, that we don't screw up, even with fetal calf serum or even with 100 days of culturing. Those are hardwired capacities, and now the challenge is for all the companies to learn how to use them in the most efficacious way.
But do you think that the different processes, because all those companies got different ways of actually selecting them, growing them, you know, all the steps that potentially changes it, but they do retain that vital thing. But do you think they have different proportions? So maybe the one from company A has much more angiogenesis capacity, company B maybe has much more, you know, immunomodulatory capacity. So they tune, or you can tune the various capabilities of the different cell lines for different indications.
I think I would ask a different question, Chris, which is, all of these preparations are going to be intrinsically different, as you point out. The real issue is, what are they doing in vivo?
Absolutely.
And so the question you're asking is there a way of stoking the furnace in the MSC for heart versus kidney, for heart versus stroke? And the answer is we don't know. And so you know, I have to say, this is hard for me to say, is when I lecture to medical students, I talk about 100 years ago, we ground up dog pancreases and gave them to diabetic patients, worked fine. Then we became sophisticated and we got purified pig insulin. And then we did human recombinant insulin. And now actually Mike Weiss at Case Western Reserve has fiddled with the amino acid sequence of insulin, and so he can get it actually to not be degraded in the bloodstream for 3 days. So this is really high tech now. And where are we with MSCs? We're grinding up dog pancreases. But it works clinically. And I think we need these clinical trials, and we need this clinical data. And again, the safety profile for these cells are as far as we know, quite excellent. And all of the clinical models we use in my lab for asthma, for cystic fibrosis, for MS, for inflammatory bowel disease, for all of these rodent models, we use human culture expanded MSCs. So that curtain works in rodents. So it's a very, again, this is very powerful stuff. And now for basic scientists like myself, the object is, now that we put human MSCs in rodent disease models, we have an opportunity to try to interrogate the human cell to find out what human molecules that cell's making by laser capture. And actually the more interesting and unforeseen piece, which is obvious once I say it, is the tissue sitting next to an MSC itself is making a different spectrum of proteins than the injured tissue that doesn't see an MSC. So the question is, who's making the therapeutic proteins? Is it the Good MSC, or is it the host?
Or his friends next door?
My thesis is that MSCs manage the host's regenerative capacity. And so therefore, our bet, and we have some data to back this up, the host is making the therapeutic molecules. So the issue is, how many MSCs do you need at a site of repair and regeneration? Our view is tens to hundreds.
So small numbers. So manufacturing might, because 10 to the 9 is some of these therapies at the moment.
Problem,
big numbers.
Well, the problem with, I just saw my good friend from Lonza in the back, who produces gazillions of cells and has wonderful activities, wonderful facilities, and Bob Petrie also has fabulous facilities for enormous culture expansion of these cells. We're lousy at delivering them. We deliver 1 to 5 million cells per kilogram when hundreds are therapeutic. So the challenge for tomorrow, and the challenge for all the entrepreneurs in the audience, is how do you deliver them more precisely in a more efficacious package? And then your comment is exactly the next step, which is how do you prime them for that site to be the most efficacious? So we're grinding up dog pancreases. We're at that stage. It kills me to say that because I want to be so sophisticated, right? But the truth is, the science and the entrepreneurial activities of the people in this room are necessary to perfect this science. We need to get this therapy to the people who could use it, and so it's going to be at the current way clinical trials are done, at 1 to 5 million cells per kilogram. But 5 years from now, 10 years from now, we need to change this, and we need to be smarter about how we deliver the cells, and in your context, how we prime them for that site. So we're at a very crude stage at the moment, and again, evidence-based clinical trials, obligatory, fast tracking, not such a bad idea. And I just published a paper.
Can I just pick up something, because we've got to close soon.
Sure.
One last bit, which I think is really what people would like to hear from you as the person who originated the Caplan cells, let's call them in this thing. But they have been immensely successful. 400 clinical trials, masses of research papers, whole careers built on them. If you add up the market caps of the companies there, it's probably about 2 billion. That's a tremendous record. But where is it going to be over the next 5 years? Where do you think the success of MSCs is really going to be?
I think 2014 is the breakout year for cell-based therapy. And I think that you're going to see data from these clinical trials. Mesoblast, for example, just published in January, phase 2 trial squirting MSCs into spinal discs. Yeah. The MSCs do not differentiate into disc tissue. They make drugs, and it's the disc itself which is curing the patient. And the clinical data that they have is spectacular. So again, this is medicinal. 5 years from now, in rural England, John Smith is going to have a heart attack, and the helicopter's not going to get him and take him to a London hospital. He's going to get in his car and go a mile down the road to an urgent care center, and from the freezer, they're going to take a bag of cells and hook them up to an IV, and that's going to be the therapy. And I think that if you just think about the economics of that, if you think about if these cells really do work in AMI that easily, it's going to be a product that's going to sell like wildfire. And again, if I had MS, if I had ALS, and there's fabulous clinical trials. We just finished a clinical trial in Cleveland, investigator-initiated clinical trial on MS. If I had MS, I'd be shooting up. And you can't do it in England, and you can't do it in the U.S., and there's lots of negativity amongst us with regard to medical tourism. I think countries are cleaning up their act, but if these cells can really, I want to make one very important point, because there's some regulatory people here. So don't subject MSCs to all the failed therapies. So if a patient fails all of the 52 drugs that are available for MS, and then you give them MSCs, it's the wrong patient. The patient who's newly diagnosed with MS, who's newly diagnosed with ALS, the kid who is getting diabetes, those are the patients who should be getting MSCs, and they need them now. And so if we can get the data to prove that, we're going to need the regulatory community to cooperate with us and not give us all the failed cases so that we can try our products on those cases. It's illogical, and also I think it's unethical for the physicians to be doing that.
A great final comment for discussion over the coffee break. Thank you very much, Arnie. Great, seriously.